Photosynthesis and Cellular Respiration by Benjamin Charbonneau and Rowan Group. Photosynthesis. It all starts with a plant. Inside that plant there are cells, and inside each cell there are mitochondria, a nucleus, chloroplasts, and the cytoplasm. An actual plant cell wouldn't look like this. It would look more like this. As you can see here, all the different parts of the cell are labeled. There's the mitochondrion, there's the chloroplast, there's the cytoplasm, there's the nucleus, but as you can probably see, there's a lot more. I've just covered the basics of this plant cell, but if there's anything else that I haven't mentioned that is important, then I may refer back to this model further in the video. Anyways, the first step to photosynthesis occurs inside the chloroplast. Inside the chloroplast, there are thylakoid membranes. The process begins inside these thylakoid membranes. Now, the process of photosynthesis is a pretty long process, and I probably wouldn't be able to green screen all of it because my phone would crash before I'd be done. So this next part I just have modeled with text. First, chlorophyll captures light energy, which then it uses to split water into oxygen and CO2. Next, inside the stroma of the chloroplast, the enzyme ribosco takes CO2 and combines it with a 5-carbon sugar called ribulose 1,5-biophosphate, or RUBP, to make a 6-carbon intermediate that is unstable and then immediately splits into 6 molecules of a 3-carbon compound called 3-phosphoglycerate, or 3-PGA. After this, the 3-PGAs are converted into glyceraldehyde 3-phosphate, or G3P. Next, 5 of the G3P molecules rearrange into 3 molecules of 5-carbon RUBP. The next step, glycolysis takes place inside of all cells, animal and plant. Glycolysis takes place inside the cytosol of all cells. First, hexokinase uses 1 ATP to convert glucose into glucose 6-phosphate, or G6P, trapping it inside the cell. Next, phosphoglucose isomerase converts G6P into fructose 1,6-biphosphate, or F1,6BP. After that, analase splits F1,6BP into dihydroxyacetone phosphate, or DHAP, and a glyceraldehyde 3-phosphate, or GAP. Next, triphosphate isomerase converts GHP into another GAP. After that, glyceraldehyde 3-phosphate dehydrogenase, or GAPDH, oxidizes GAP and adds an inorganic phosphate, which makes 1,3-biphosphoglycerate, or 1,3-BPG, and NADH. After all this, phosphoglycerate kinase transfers a phosphate from 1,3-BPG to ADP, forming ATP and 3-phosphoglycerate, or 3PG. Next, phosphoglycerate mutase moves the phosphate on 3PG to form 2-phosphoglycerate, or 2PG. Then, analase removes water from 2PG to create phospho-N-pyruvate, or PEP. Finally, pyruvate kinase transfers the phosphate from PEP to ADP, yielding ATP and pyruvate. Basically, in the end of glycolysis, 2 pyruvate, 2 ATP, and 2 NADH molecules are formed per 1 glucose. The next step, which also takes place inside all cells, is the Krebs cycle. The Krebs cycle begins when a cellular CoA, or 2C, combines with oxalate, which is 4C, to form citrate, which is 6C, catalyzed by citrate synthase. Next, citrate is isomerized into isocitrate, 6C, by acetylase. After that, isocitrate is oxidized by isocitrate dehydrogenase to form a glutarate, or 5C. This releases CO2 and produces NADH. After that, A-ketoglutarate is oxidized to form succinyl CoA, or 4C. A second CO2 is released, and another NADH is made. Succinyl CoA is converted to succinate by succinyl CoA synthase, producing ATP. After that, succinase is oxidized to form 4C by succinate dehydrogenase, producing FADH2. Next, fumarate is converted to malate by hydration using fumarase. After that, malate is oxidized by the malate dehydrogenase to regenerate oxaloacetate 4C, releasing the final NADH. Next up is called the electron transport chain. This occurs inside the inner mitochondria membrane. First, NADH drops off 2 electrons at complex 1, becoming NADH+. Next, complex 1 uses the electron energy to basically move protons into the inner membrane space of the mitochondria. After that, FADH2 drops off electrons at complex 2, becoming FAD. Then, both complexes 1 and complex 2 pass their electrons to their mobile carrier. Uniquelone then moves to complex 3, which pumps 4 more protons across the membrane. After that, electrons move from complex 3 to the second mobile carrier. Then, cytochrome C delivers electrons from complex 4, which pumps the final 2 protons out. After that, oxygen wakes at the end of complex 4 and accepts the spent electrons. Then, oxygen combines with nearby protons, which H+, to form water, H2O, as a byproduct. Then, a high pressure concentration of protons builds up in the inner membrane space of the proton motive force. Protons flush back into the mitochondrial matrix through the ATP synthase enzyme. The flow of protons causes the internal axle of the ATP synthase to spin mechanically. This mechanical energy basically forms a phosphate group of ADP, which then basically creates ATP. This process generally creates about 32 ATP after 2 cycles.